Hi, I'm Sharon King. I'm a senior scientist at St. Jude Children's Research Hospital. I'm part of a microscope core facility in the developmental and neurobiology department, and I provide access and support for biologists that are studying developmental neurobiology. I'll be talking today about quantitative phase imaging by presenting you a variety of imaging methods that are transmitted light methods as opposed to fluorescence imaging methods. My intention and my hope is that you'll become familiar with ways that you can introduce contrast in a sample, reveal interesting biology, but without using fluorescent labeling techniques. So the great advantage of transmitted light methods is that they require so little illumination. Intensity levels needed to capture high contrast images are so low that they can be used to image live samples in a microscope environment with very low levels of phototoxicity and without potential functional interference from fluorescent labeling complexes. So from this perspective, transmitted light microscopy is ideal for studying the dynamics of biological systems. So you can imagine imaging for hours or maybe days, or as long as you can keep your model system going, whatever model system that might be. In the past, fixation and staining methods were required to define clearly what was being observed in transmitted light imaging because the contrast is nonspecific. And so studying biology, especially on a submicron scale, has historically best been done using fixed samples. It's my opinion that applying transmitted light methods in combination with live cell fluorescent methods is a very powerful way to derive information about specific fluorescently labeled submicron dynamics, for example, GFP gene expression markers, and the morphological subcellular context surrounding those systems, which might be, for example, the formation of synapses or the movement of filopodia. The development of transmitted light microscopy methods that are quantitative are often referred together in one category called quantitative phase microscopy. And that development has created a variety of systems that provide submicron resolved 3D volumes of refractive index and material density information. So transmitted light methods can now quantify optical properties of the sample as well as provide qualitative contrast. On their own or paired with live cell fluorescence, you can use transmitted light greatly and to extend both the limits of temporal resolution and the longitudinal limits of time-lapse microscopy. The main thing I hope you'll be able to take away from this presentation is that transmitted light imaging methods are ideal for live and quantitative microscope applications. Typically, transmitted light imaging means illuminating the sample with a source that's orthogonal to the view of the sample, and that's the case in bright field microscope imaging. Contrast in bright field is created by absorption of the transmitting light by the object, and in this way, the intensity in the image represents absorption. So most structure in typical biological samples is too weakly absorbing on its own to introduce much contrast. On the left, for example, are unstained epithelial cells that are very difficult to see. So commonly introduced in are absorptive stains, as in the case of this array of plugs of tissue sections that have been stained using common pathology methods and hematoxylin and eosin stains. Stains are typically used on fixed samples, and stains of any kind are problematic even when applied to living material. In a bright field image of a fixed sample, the changes in intensity that you see are created by changes in the absorption properties of the chromophore, and the intensity may not be a linear function of absorption. Absorptive chromophore stains are generally not quantifiable in the sense that the amount of absorption is related only to the amount of dye and its properties, not necessarily to the biology that's interacting with the dye. The phase contrast and polarization methods that I'm going to talk about for the largest portion of this presentation use optical elements to create contrast rather than stains. So now imagine that this sample is illuminated with a high angle of incidence relative to it rather than orthogonally. Specifically at an angle of incidence that's higher than can be transmitted undeviated through the imaging aperture. This method is called dark field microscopy because in the background of the field of view, where there is no sample and in which no diffraction is taking place, the image is completely dark. Only where the sample alters the direction of light, either through diffraction or scattering, can the light actually be collected into the objective and imaged. To create this type of contrast, the transmitted light microscope usually requires a specialized condenser housing that has a combination of mirrors to create a very high angle illumination such that the undiffracted light bypasses the maximum angle collected by the highest numerical aperture objective. This specialized type of condenser is not entirely necessary though, because it's also possible to create a dark field effect by pairing a lower NA lens with a large annulus in the illumination path. So you can use a traditional condenser lens without these mirrors, and those traditional condenser filter wheels will have annulus filters that are meant for use with phase contrast microscopy, which I'll talk more about later. So all you need to implement a low NA version of dark field is an annulus that has an inner diameter larger than the back aperture of the imaging objective you plan to use. Here you see images showing a comparison of bright field on the left and dark field on the right. The top is a sample of algae and the bottom is a sample of a brine shrimp to give you an idea of the difference in contrast between these two modes of imaging. Notice how the edges of the high contrast and dark field show up very nicely, and how the microbubbles that are in the media, which are almost impossible to see in bright field, are very obvious in the dark field image. Another comparison of bright field on the left and dark field on the right. These images show sections from a zebrafinch bird brain in late embryonic stages. Here in this study referenced at the bottom of the slide, the authors labeled the expression of D1B, which is a dopamine receptor, using an in situ hybridization method with radioactive nucleotide markers that could be coated with a silver emulsion over the tissue. And that emulsion became very strongly absorptive wherever the protein expression was taking place. So in the upper left image at embryonic day 10, where the genetic expression of the protein is low, the absorption of the emulsion is barely visible in bright field. But in the dark field, it shows up well. And likewise, the expression of a SLT3 protein seen in embryonic day 12 in the lower left image and the lower right image show you how the comparison of contrast in the dark field image with the absorption of crescent violet counterstain in the bright field image can give you more information about where these proteins are being expressed, more than you could see with just either method alone. The advantage of dark field is its sensitivity to scattered light. And while it's not a quantitative imaging method in the sense that image intensity can't be directly analyzed, it does provide the potential to observe very small objects with very high contrast as long as they are scattering. Even objects that aren't necessarily optically resolved but scatter at high angles are detected and observed. An example is a study of 25 nanometer diameter microtubule dynamics. I'm showing an example image of that here in the upper left. Another example, the dark field imaging method is excellent for capturing low magnification outlines of individual cells, shown on the bottom right. Instead of trying to use a fluorescent membrane marker, which might be difficult to find for certain cell types, or maybe following a dynamic cycle of its own, making it difficult to use as a reference, dark field can be used to show the outline of cells. This is an example of a microscope method that has the potential to produce very high contrast images without any stain at all, and it's often overlooked. But it can be an excellent way to observe dynamics of live cell samples with very, very low amounts of illumination. Same can be said about all the transmitted light imaging techniques I'll talk about, but particularly in dark field, the contrast is high and the illumination can be even lower than for most other transmitted light imaging modes. Phase contrast microscopy is also a great way to observe live cell dynamics without stain by converting the phase delay of an object into an amplitude contrast. So the phase delay of an object is the difference is created when there is a difference in refractive index between an object and its surrounding medium. And that object causes light to slow down with respect to the background. These two images show you epithelial cells, and on the left is the bright field image, and on the right-hand side is the phase contrast image. The phase contrast method was introduced by Fritz Zernicke in the 1930s, and is the method you'll most likely find on a microscope that's set next to a culture hood, for example. However, there are many phase contrast methods, and now it's a more general term. Over the past 80 to 90 years, there are many ways of introducing phase contrast. And so I'll talk about these additional methods as phase contrast as well. But I'll try to refer to the original as Zernicke phase contrast. So Zernicke phase contrast requires additional optical components to the bright field microscope. There will be an annulus in the illumination path such that the middle of the aperture of the condenser lens is blocked. The image on the lower right here in your slide shows the filter wheel that sits in front of the condenser lens. This has different size rings for each objective, usually labeled pH 1, 2, and 3, and the rings are sized to match a ring of quartz that sits in the back aperture of the objective housing. You'll find that phase objectives are usually labeled on the outside with the size of the matching illumination ring, conventionally in green colored markings. A phase objective is entirely transparent and it contains a ring of quartz within the glass. And the material has a refractive index and a thickness such that the light passing through the phase ring is delayed a quarter wave with respect to any light that's passing through the rest of the glass portions of the objective. So the effect of the plate in the imaging objective is that the amplitude of light waves reaching the detector will sum together constructively to give an increased contrast between the sample and the background. Whether the sample appears darker or lighter than the background will depend on whether the phase ring is engineered to advance or delay the phase of the undeviated wave relative to the deviated wave. The undeviated wave or undiffracted wave is the light that doesn't interact with the sample. You may hear terms positive or negative phase contrast to refer to the two possibilities of a light or a dark background. Most common is positive phase contrast, and the image examples I'll show will all be of positive phase contrast. The diagram on the right here shows the optical path of the two waves. The red path is the path of the illumination. A ring of light comes from the annulus filter and will pass through the condenser lens and evenly illuminate the sample with a set of high-angle plane waves, which, in areas where there's no sample, will travel undeviated through the correctly sized phase ring at the back aperture of the objective. Light interacting at any position with the sample will be delayed or diffracted due to the thickness and refractive index variation of the sample. So what we observe in the image plane will be a sum of the transmitted light waves, deviated and undeviated. On average, you'll have a delay from a typical sample that will be on the length of a quarter wave, about 200 microns. A quarter wavelength lag produced by a sample, plus the quarter wavelength jump given by the background light, will give you a total of a half wave shift, which is ideal to produce high contrast interference. So the plot on the upper left here shows you an example of the wavefronts approaching the detector without the phase plate. In that case, the undeviated wavefront and the deviated wavefront sum together to create a resultant wave, which without the phase plate is relatively low contrast. Whereas if the two beams are brought into phase with each other, as shown in the plot on the lower left, the image contrast is high. So the three transmitted light imaging modes I've presented to you so far all produce very different types of contrast. So it can be kind of fun and informative to compare the images of the same sample in all three modes. So for example, in this cell, it's possible to see absorption by vesicles along the outer edge of the nucleus in the bright field image, while in phase contrast, the full shape of the outer membrane is very clear, as well as some specific regions in the lamellipodia and inside the nucleus. In the dark field image, what we see is a bright scattering by subcellular organelles located around the outside of the nucleus. Analysis and interpretation of phase contrast images, whether they're created using Zernike phase contrast or any of the other phase contrast methods I'll speak about, require keeping in mind the particular method of contrast formation. The difference between positive and negative phase contrast is an example. You can consider the product of the thickness of the sample and its refractive index to be the phase of the cell, since this is the value that delays the wavefront. It's also a measure of the dry mass of the sample and is therefore sometimes referred to as the optical density of the sample. In these images of yeast, you can see that the yeast cells appear dark against a light background, indicating they're denser than the surrounding medium. There are also lighter spots within the yeast and a bright line across the zoomed up yeast cell that indicate there are areas where the density of the object is low and close to the surrounding medium. However, you can't interpret whether this is due to a change in refractive index or a change in thickness, unless you have some kind of additional information. Finally, in Zernike phase contrast specifically, because the diffracted light passes through the whole objective and some portion also passes through the phase ring, there's often a halo artifact, especially around the edges of an object. And this can make both qualitative and quantitative interpretation of these types of phase contrast images complicated. For instance, in these images, is the line across the cell real or is it an artifact? The halo effect is, in my opinion, the worst drawback of Zernike phase contrast, and otherwise its disadvantages are very slight. It has some very important advantages over other phase contrast methods. The most important is that it's compatible with plastic cell culture dishes. Traditionally and most commonly, phase contrast is a qualitative imaging method. Its intensity is not linearly related to the phase of the object. However, it is possible to apply phase shifting and computational image reconstruction methods to extract quantitative information from these types of phase contrast images. So one commercially available approach for extracting quantitative phase from phase contrast images is provided by the company Phi Optics. And it uses the principles of Zernike phase contrast, but rather than capturing a single image with a static phase delay inside the objective, this configuration, called spatial light interference microscopy, uses a lens relay to create a plane conjugate to the back aperture of the imaging objective outside the standard commercial microscope stand. The ring aperture in the phase objective is replaced in this relay with a liquid crystal spatial light modulator that imparts a ring-shaped phase delay pattern that matches the condenser annulus in the illumination path. The liquid crystal spatial light modulator enables a series of phase images to be captured in which the amount of phase delay in the ring can be shifted by known amounts and at high speed. The liquid crystal modulator can be programmed to implement a ring appropriate to any imaging objective NA. So the optical properties of the liquid crystal are similar to those of uniaxial crystalline materials and are similar to what is used to create static wave plates in traditional phase objectives. Each pixel in the modulator consists of an array of long molecules, the liquid crystal molecules, whose orientation with respect to the propagation direction of light will change the refractive index property of the pixel. And in a liquid crystal spatial light modulator, this orientation is electronically controlled, and so variations in refractive index can be calibrated to create specific phase delays that are synchronized with the detector, and you can capture 3 to 4 images at rates of around 100 Hertz. These 4 images of a neuron, for example, are then used to compute a final result in which the intensity is quantitatively mapped to the object phase. Thanks to the speed of the phase modulator and graphics processing computing power, it's possible to capture the images at rates that are applicable to time-lapse experiments. Final result of the image reconstruction extracts only the information related to the phase of the wave at the detector, which is shown here in this image of a neuron as a heat map of phase values. This slim method is able to quantify very small variations in phase down to the nanometer scale. Remember that phase delay is imparted by the object, and it's a value that's the product of refractive index and thickness. In this approach, where the sample is illuminated uniformly, that phase information is averaged over a volume. The volume is defined laterally by the diffraction limit of the numerical aperture of the objective and axially by the depth of field of that objective. Practically, what that means is that since the higher the resolution limit of the objective, the shallower its depth of field, this type of approach works best when applied to thin samples like these neurons cultured on glass. Some pros of this slim spatial light interference microscope method are that it adapts to any research-grade commercial microscope as an add-on, and of course can still be used with plastic dishes. It uses an incoherent halogen lamp light source that's cheap and produces high contrast images, but unfortunately it's still subject to produce the halo artifacts of Zernike phase contrasts, the image formation method that it's based on. And these artifacts are especially noticeable when imaging objects that are thick compared to the depth of field of an objective. And while this method negates the need to buy phase contrast objectives, which can be expensive, the cost is swapped for the cost of a liquid crystal spatial light modulator, which is also expensive. A phase contrast method that's not expensive and is still very useful is Hoffman modulation contrast. It's sometimes called poor man's phase because it can be implemented with very cheap materials, but it can also produce very high resolution phase images without those halo artifacts of Zernike phase contrast. And it's heavily relied on in vitro fertilization labs where cells are cultured in plastic dishes routinely. So for example, these are images of oocytes. The top image is taken from a Leica webpage advertisement of an integrated Hoffman modulation contrast digital microscope laboratory, specifically certified for in vitro fertilization work. The principles of this Hoffman modulation contrast method are very similar to those of phase contrast and dark field. Rather than using a ring annulus, the illumination has a slit filter in front of the aperture of the condenser that directs the high angle oblique illumination onto the sample. The undifracted background light passes through a matching modulation filter in the back focal plane of the objective. However, this second filter in the back focal plane of the imaging objective is usually outside the housing of the lens and used with a low numerical aperture lens whose back focal planes can be accessed. The matching strips in the modulators are designed to transmit 15% of the background light, and that reduces the background to match the light intensity of the diffracted light. The largest area of the matching filter blocks 100% of the background illumination while passing 100% of the transmitted light. The smallest region transmits 1% of the undifracted illumination. So in order to observe contrast in this method in the imaging plane, the sample has to divert some light through a combination of either diffraction or refraction through the portion of the upper modulator that transmits 100%. The advantage of this phase contrast method I've already mentioned, but its disadvantages are that it has a directionality to the contrast, and there are light and dark shadows that give a pseudo 3D appearance that can lead to confusion in some samples. And with samples that have linear structures, this orientation of the imaging contrast will cause the contrast of the structure to fade and disappear if it's parallel to the length of the illumination slit. Typically, this Hoffman modulation contrast is used to identify a phenotype. That is a set of observable characteristics that indicate a healthy embryonic development. So rather than being quantitative in terms of intensity, it's quantitative in terms of phenotype. Another interesting method that isn't exactly a transmitted light method, but draws on many of the same principles I've just presented, is called interference reflectance microscopy. And it's a method for creating contrast without staining, so it's ideal for live cell imaging, same way that all the methods I'm presenting are. And in this type of configuration, you've got high angle illumination once again, and it's used to illuminate, but this time the objective serves as both the condenser and the imaging lens, and light is reflected from any interface where there's a change in the refractive index. That's at the cover slip to medium interface, the medium to cell interface, or if the cell is adherent to the glass, then the glass to cell interface. Reflected light is imaged through a partially transmitted mirror to the detector, and reflections from different interfaces travel different path lengths that are dependent on both the distance between surfaces and the difference in refractive index between the surfaces. In other words, there's a phase shift between the beams reflecting from different interfaces. These phase shifts cause constructive and deconstructive interference at the detector that can be observed as differences in intensity. The addition of a polarizer in the illumination path and an analyzer in the imaging path can be used in combination with a quarter-wave plate to enhance the contrast of the reflected beam and to tune the contrast to a specific interface. For example, usually you want to observe the biological sample interface with the glass surface. Here, for example, are images of phase contrast and interference reflectance microscopy in comparison. Reflections off of biological interfaces are very weak, and most of the illumination is lost to transmission. However, the optical enhancement that can be done with polarization control or with computational enhancement through digital filtering can increase the image contrast and allow you to observe things that can't be observed with other methods. In these two images, these are glioma cells. The phase contrast mode on the left and the interference reflectance mode on the right appear very different. There are attachment plaques where the cell meets the glass, and these are clearly visible in the right-hand side in the interference reflected image. While on the left-hand side, the plaques are invisible, but the nuclear membrane and internuclear subcompartments are seen, which are only seen in phase contrast. So in some applications, the reflections off of an interface are strong enough to make observations without polarization control or digital enhancement, but that will depend on the sample and the degree of coherence of the illumination. So notice that the primary reflections here are at the interface between the cover glass and the cell, and they're due to the relatively large refractive index difference between them. The depth from the cover glass at which contrast can be produced using this method is shallow, and it's well aligned with the depth of information that's obtained from total internal reflectance fluorescence, which is also extremely shallow. In this example, COS7 cells were stained with a biotinylated antitubulin primary antibody, and that was dual tagged with a streptavidin Alexa 647 and Alexa 488. So shown here are images at the left of fluorescence in wide field in those two colors using total internal reflection fluorescence. And while the upper right image is an interference reflectance image of the same field of view. In this case, the authors were using this interference reflectance microscope image to verify their quantitative analysis of their dual-labeled turf, which is shown in the color map views. But you can already see by interpreting visually the interference reflectance contrast, this cell is detached from the glass on the right-hand side and is strongly adhered to the cover glass on the left. On the left of your slide, I've listed some more interesting applications of interference reflectance microscopy. It's a unique method for visualizing surface properties of unstained cells in a way that can't be used or can't be seen using transmitted light. And like all these methods, it's stain-free and great for imaging live cell dynamics with very high temporal resolution. Of course, it's limited to reflective surfaces that are within the wavelength of illumination. And usually that means the first reflection from the non-glass interface is the only one that can be resolved. And while it's typically used and interpreted in a qualitative way, the number and shape of the interference fringes that you see can be used for a quantitative analysis. And it's been demonstrated that it's possible to derive quantitative topographic information from sequentially illuminated multi-wavelength interference reflected microscope images. Differential interference contrast is an interferometric transmitted light phase contrast method rather than reflected. Because it uses polarized illumination, it can't be used through plastic dishes or flasks because plastic depolarizes light. And in those cases, Wernicke phase contrast or Hoffman modulation would be a better choice. But differential interference contrast produces a better quality phase contrast in thicker samples than those methods, and when high resolution is needed because none of the aperture is blocked in this method. So it makes full use of the numerical aperture of the imaging system. These two images shown here capture Glioma cells. On the left-hand side is the bright field image, where it's almost invisible, and on the right-hand side is the high-resolution differential interference contrast image. The light path of the differential interference contrast system also has several additional components compared to the bright field microscope. Besides the polarizer and analyzer that you would use in polarization microscopy, there is also a beam splitting and a beam combining prism called the Wollaston or Nemarsky prisms. The prisms are composed of calcite, pieces of calcite, which are glued together and combined with their optic axes in specific orientations. The input illumination is linearly polarized at 45 degrees to the eigenpolarizations of these prisms, and the lower prism acts as a beam splitter, while the upper prism is a beam combiner. After the beam splitter, the two beams pass through the object sheared apart along a single direction called the shear direction. The beams are recombined at the upper Nemarsky prism, pass through the analyzer and to the detector. The analyzer is a linear polarizer that's crossed with respect to the illumination polarizer. And contrast is produced through interference of the orthogonally polarized beams that are separated or sheared laterally by a distance that's smaller than the resolution of the imaging system. The intensity distribution is changed by laterally shifting the beam combining prism to add a phase bias to the interference pattern resulting in the detector. So for example, in this DIC image, the intensity distribution reflects the difference in refractive index with respect to the background of a polystyrene microbead, and the addition of a phase bias is due to the lateral position of the prism and the relative phase delay of the two polarized beams. In this configuration, you can see the relationship of the intensity in this imaging mode to the differential phase of the object, from where it draws its name, DIC. Removing the phase bias changes this intensity distribution in such a way that the edges of the bead become preferentially brighter in a similar way to the dark field imaging method. Or greater phase bias could be added to invert the contrast of the differential intensity. When the phase bias is set such that the background is in the middle of the contrast range, the intensity in a DIC image can be thought of as related to the derivative of the optical path delay of the object, as shown here using the example of a blood cell. The refractive index and thickness of the cell, remember, imparts a delay in the optical path of the transmitted wave that's relative to the background, and that is represented by the line plot in the top center here. The derivative of the line plot at the top right is representative of the amplitude observed in the DIC image along the shear axis. The effective phase bias is shown in these plots with the example of droplets. You can see that this derivative-like effect is due to the differential subtraction of the polarized wavefront phase shifts and the effect of the phase of the amplitude on the final interference pattern. In general, the image of the sample will, like Hoffman modulation, have a 3D-like appearance, with the features protruding out of the plane or into it, depending on whether you're using negative or positive phase bias. And the position of no phase bias, it's called extinction, looks a lot like dark field. For any particular sample, the user shifts the phase bias around to optimize the contrast and will typically attempt to show their structure of interest to something that pops out of the display like a hill rather than into the page like a divot. The key advantage of this phase contrast method over others is that it can be used with high numerical aperture objectives, and it provides excellent lateral resolution as well as very thin optical sectioning in relatively thick samples without producing artifacts. But its key disadvantage is that its use of polarized illumination makes it incompatible with plastic dishes or incompatible with any intrinsically birefringent sample. The optical principles of DIC can be used in a way similar to the method of SLIM to capture a series of images with a known phase bias and compute quantitative phase image. The series of images might be a set of images that have known phase shift between them, such as these images of a nematode, or they may be a set of images that are acquired at different angles of rotation relative to the camera, or some combination of both. The quantitative phase result is computationally extracted from the series. So for example, the top row of images here is a set of phase-shifted images of a nematode. And these images, plus a second set of 4 at 90-degree rotation, were used to compute a quantitative phase image, and the phase contrast there is linearly proportional to the phase. And then separately, it's possible to compute the amplitude image, where the contrast is due to absorption or loss due to scattering. In phase microscopy generally, a linear relationship between intensity and phase, which is the thing you want to quantify, doesn't exist. So instead, the relationship between the object and the intensity is more complex. It's a result of both absorption and diffraction. And all quantitative phase microscopy methods use some approach to restore a linear relationship between the image intensity and the object phase. Keep in mind that the phase contrast in the quantitative phase images still does not specifically label any biological structure. The contrast is a direct consequence of the interaction of the illumination with the physical properties of the sample. Dr. Michael Schriebach, who is a research scientist at Woods Hole Marine Biology Lab, has developed a liquid crystal technology that can be used to replace beam splitting and beam combining prisms of the DIC microscope. In this way, DIC images can be captured with any orientation of shear, and pairs of images with varying shear orientation can then be used to compute quantitative phase images. This image was acquired using Dr. Schriebach's orientation independent DIC with the tunable prism replacements. And it's a movie of a small wound in the epithelium of hydrozoan something. Anyway, this imaging system is also capable of imaging time-lapse with millisecond exposure times and correspondingly short intervals. You can see in the video contraction of the cell edges and the change in shape of the adjacent cells. As a quantitative phase microscope approach, DIC-based quantitative phase takes advantage of all the same advantages of qualitative DIC and all the same disadvantages. But as far as I know, it's still under development in the lab rather than being commercially available. A commercially available approach to quantitative phase that is provided as a microscope add-on device comes from a company called Phasix. And this approach uses a method referred to as Quadrowave lateral shearing interferometry. In this method, what is recorded is not the image directly, but a type of interferogram that's created by placing a mask in front of the camera. The mask can also contain an array of lenslets. Either way, it measures the local tilt of the transmitted wave by detecting lateral shifts in intensity transmitted through the mask. Whereas an undeviated beam would transmit straight through the mask, a deviated beam will shift the transmission of the mask. This is equivalent to measuring the phase gradient of the sample-induced phase shift. This approach to phase measurement is derived from the Shack-Hartman wavefront sensor. If you have some familiarity with that device, it's often used in measuring the beam shape characteristic of laser outputs. The output in this case is very closely related to that of differential interference contrast because it produces intensity that measures phase gradients in a particular direction. The DIC-like images that it produces are computed from a single acquisition with this type of device, and it demonstrates that you can capture the phase gradient in two orthogonal directions simultaneously. Computational phase image reconstruction process from lateral shearing interferometry requires that you demodulate first. The interferogram that you capture is filtered in the Fourier domain or spatial frequency domain to extract only the object information, which in this case is the phase gradient rather than the phase directly. The phase gradient is the derivative of the function that represents the phase delay, and this step produces a set of intermediate images that appear very similar to DIC images Not surprisingly, because DIC is also a lateral shearing interferometry method, but if we called it that, we would have to call it uniwave because it occurs only along a single direction. So a significant difference between this approach and DIC is that quadrowave lateral shearing interferometry doesn't depend on polarization control to create this interference, and is therefore not distorted by polarization dependent properties of the sample. So it can be used with plastic dishes. The phase gradient information computed from quadrowave lateral shearing interferometry is combined and integrated using numerical integration approaches. One such approach is spiral phase integration. Spiral phase integration solves the issue of streaking that's seen in line-by-line summation approaches and can be used to integrate phase gradients with very few artifacts, regardless of what symmetry the object has. This spatial integration approach can be viewed as a filtering method from the point of view of Fourier theory. And in this case, that filter would be a step function of phase in all directions, and it takes its name from its spiral shape in 2D. The phasic system has been used to establish a characteristic refractive index of lysosomes, for example, in COS7 cells, by correlating the cell vesicles with the fluorescent tag. Establishing this type of correlation between fluorescence and quantitative phase allows you to track vesicles without using a fluorescent marker, which can be a way to observe their behavior in a more natural state without any fluorescent stain. Because the segmentation of the relative refractive index distribution isn't straightforward, it's not a straightforward analogy to segmentation of fluorescence images. The use of fluorescence overlay as a validation step at this stage in the development of quantitative phase imaging is a particularly powerful avenue for developing new methods and specific applications of quantitative phase imaging. Quadrowave lateral shearing interferometry is a great method for quantitative phase microscopy. One con of this method is that the mask is specific to each objective, so that each objective requires a unique mask or a unique lenslet array designed with its resolution matched to the resolution of each objective. Still, the most common and commercially developed quantitative phase imaging available for use in biology is the approach of digital holographic microscopy, sometimes combined with tomography. The digital holography approach enables live cell quantitative phase imaging using a single image capture rather than a acquired or computed image series. In holographic configuration, the microscope is introduced into the object arm of the optical interferometer. The interferometer has two light paths split from the same coherent source. That is to say, this microscope method uses a laser rather than an LED or a lamp source. The two paths are an object path and a reference path, and these beams are recombined and interfere both constructively and deconstructively to form an intensity pattern that is determined once again by the difference in path lengths traveled. In this way, the phase of the object is encoded in that interference pattern. If encoding the phase of the object in an interference pattern still seems a little bit abstract, remember that intensity variations in a phase image occur because waves of light add constructively and deconstructively, as you see in this illustration, depending on the relative delay between the wave peaks. In phase microscopy, a phase shift is introduced by the phase of the sample relative to the reference wave. All the phase microscopy methods that I've presented so far use interference in some form or another to create intensity contrast. In holography, however, the coherence of the laser illumination allows the wavefront to retain the encoded phase in the intensity pattern while propagating over a long distance. This is different from the systems that I've presented so far that use a partially coherent LED or a lamp source. In those low coherence illumination methods, the phase of the waves quickly becomes uncorrelated after propagating only a relatively short distance and blurs out any sample encoding phase shifts. For this reason, in all the methods presented up until now, the camera has been placed such that it images the intensity at the sample plane, where the short moment in time and space of phase encoding can be captured. In holography, however, the image sensor is placed such that it captures the intensity after it's propagated to the far field of the microscope objective. Instead of recording the image of the sample, the system records the interference of the reference wave with the far field diffracted pattern of the sample, which is a hologram. There's a Fourier transform relationship between the object phase and the hologram. Phase image is numerically reconstructed, and any plane within the object can be observed in focus. Commercially, Nanolive and TomoCube are the only two companies worldwide offering a digital holotomography product. There are other companies offering digital holographic phase microscopy without the tomography for more general research microscopy. But all of these companies' products are based on essentially the same optical configuration for coherent interference, which I've been showing you. And in the following slides, I'll show a variety of examples from these commercial systems to give you an overview of the interesting applications that are out there for digital holographic microscopy with or without tomography. So this example is a quantitative phase image created using DHM from the Phi Corporation. And it shows a nice side-by-side comparison with Zernike phase contrast of cultured fibroblasts. Here you can see the halo artifacts very noticeably in the phase contrast image. And you can easily see how the image contrast on the right is much more difficult to threshold in order to identify and count cell numbers than the image on the left. Both TomoCube and Nanolive are developing tools to improve the experience of interpreting quantitative phase results. They each have a variety of tools for making color mapping of refractive index ranges to highlight the specific intensity distributions appropriately. Both companies have a library of clearly defined and validated protocols for specific applications. For example, this segmented and pseudo-colored quantitative phase image from TomoCube shows a T cell and an antigen presenting cell and captures the formation of the cell-to-cell immunological synapse. In this case, the refractive index was used to train a convolutional neural network to distinguish the adjoining immune cells, target cells, and the synapse itself. The development of that framework enables high throughput automated segmentation of thousands of immune targeted cell pairs for analysis of 3D immunosynapse dynamics, something that's not been done. You can read more details about this and this approach and its potential application in the reference below the movie. Phi Corporation also leverages the large field of view and in its system and its ability to segment quantitative phase images in order to enable statistical analysis of live cell attributes that can be measured by thresholding. So for example, area and volume distribution of cells. Being able to segment images of unstained cells opens up the possibility of adapting assays that have been developed for fluorescence contrast to images that have required no stain and no alteration of the sample. The great advantage of low cytotoxicity and phototoxicity of transmitted light methods means you can image long time-lapse series and analyze new statistics previously unavailable. Because phase information is captured in a single image without any scanning, digital holographic microscopy is really only limited by the speed of the camera. In these examples from Lindsay Tech, their product is a DHM coupled to a high-speed camera. Notice their dynamics here of the liquid lens at the left and the sperm at the right. They're captured at 100 frames per second and tracked. This is just a small sampling of the successful applications of digital holographic microscopy. There are hundreds, and new ideas are constantly being published in the literature. These images are an example from Quart Medical, which is a company which has focused their digital holographic microscopy product completely on improving the outcome of in vitro fertilization. Non-toxic, non-invasive imaging is crucial for IVF and can be used to characterize and select healthy sperm, as shown here, as well as healthy eggs and healthy embryo development. Continuing research in the field of quantitative phase imaging using DHM has also explored the possibility of improving resolution while sacrificing less in acquisition speed by applying structured illumination methods to DHM rather than tomography. Some groups that have researched improvements in the robustness of hardware and software have shown that DHM computational methods can be applied to holograms acquired with self-referencing interferometers, similar to the Shearing interferometer of DIC, and can produce high signal-to-noise holograms in very unstable environments, so that you can apply digital holography using a 3D printed holder and a laser diode and a CMOS chip, making a very cheap and portable system. This type of instrumentation can make quantitative phase accessible in a wider variety of environments where a research-grade microscope might be impractical. The reconstruction of these images can be done remotely, and the final image could be returned to the user to an off-site location. Alternatively, pseudo-color imaging of quantitative phase images can be used to allow application of traditional histological assessment protocols. Histology has been developed with decades of experience to sample preparations that are fixed and stained, but all of that experience could be applied now to samples that are not stained. And this has been shown possible using convolutional neural networks trained on histology samples to pseudo-color DHM quantitative phase images. In summary, digital holographic microscopy is a very powerful quantitative phase microscopy method and the most advanced in terms of commercial development for use in biology and biomedicine. Despite all of these advantages, the application of quantitative phase microscopy is still not well established due to the complexity and interpretation of analyzing phase images. Tomography enables separation of information due to refractive index from thickness, and the interpretation of refractive index maps still requires validation with fluorescent markers before the advantages of imaging unstained and unlabeled samples can be fully gained. The same challenges to practical biological and biomedical applications exist for all of the transmitted light and phase microscopy methods I've introduced in this presentation, as well as several that I've not mentioned that are listed here. However, fluorescent labels require many photons for excitation, either through high intensity or long time exposures, and they create a toxic environment. And it's detrimental to the long-term viability of live cells that you're trying to study. And will simultaneously inevitably photobleach with repeated exposure and no longer be detectable over long time scales. The advantage of transmitted light in quantitative phase microscopy lies primarily in the low number of photons needed to acquire high contrast, and the correspondingly low phototoxicity in the sample environment, such that time-lapse microscopy can capture dynamics of biological systems over long time scales. Thanks very much for your attention, and I hope you've learned something about quantitative phase today.